Bismillah ar-Rahman ar-Rahim. The subject of this lecture is steroid hormone biosynthesis. As we know, all steroid hormones are derived from cholesterol, and probably with the exception of vitamin D, they all contain the same ring structure, which is the cyclopentanophenanthrene ring. Also, they have the same atomic numbering like cholesterol. Just to briefly review the structure of cholesterol, cholesterol is composed of the steroid nucleus and a hydroxyl group attached to carbon atom number 3 and a side chain of 8 carbons attached to carbon atom number 17. The total number of carbon atoms in cholesterol is 27 carbons. In the biosynthesis of steroid hormones from cholesterol, the first step in this biosynthesis is the rate-limiting irreversible cleavage of 6 carbons from the 8 carbon side chain. So this produces pregnenolone, which has 21 carbons. In the biosynthesis of different steroid hormones from pregnenolone, steroid hormones with 21 carbons are known as pregnanes. Those with 19 carbons are known as androstanes. And those with 18 carbons are known as estranes. Important organs for steroidogenesis are the testis, the ovary, and the adrenal cortex. In this lecture, we'll concentrate on the steroid hormones produced by the adrenal cortex. Histologically, from the surface, beneath the capsule, we have the zona glomerulosa, followed by zona fasciculata, and then zona reticularis, and then the medulla. This is from the histological point of view. The cortex is composed of the zona glomerulosa and zona fasciculata and zona reticularis. But functionally, we are dealing with only 2 cortical zones, namely the zona glomerulosa and the combined zona fasciculata reticularis together. And there are grounds for this functional classification. First, zona glomerulosa is characterized by the production of an enzyme that's not produced by the zona fasciculata reticularis, namely the enzyme which is called aldosterone synthase. So the main steroid hormone produced by the zona glomerulosa is aldosterone, and it is not produced by the zona fasciculata reticularis because this enzyme is missing. On the other hand, 17-alpha hydroxylase is present in zona fasciculata reticularis, but it is missing in zona glomerulosa. And this enzyme is essential for the production of cortisol and androgens. So the presence of the enzyme 17-alpha hydroxylase in zona fasciculata reticularis makes it capable of producing cortisol and androgens, while zona glomerulosa is not capable of producing the same hormones because it doesn't contain the enzyme 17-alpha hydroxylase. So from the functional point of view, the presence of distinct enzymes, aldosterone synthase for zona glomerulosa and 17-alpha hydroxylase for zona fasciculata reticularis, differentiates functionally between the two zones. Another functional difference between zona glomerulosa and zona fasciculata reticularis is the fact that zona glomerulosa is controlled by angiotensin II and also by potassium ion, while zona fasciculata reticularis is controlled mainly by the hormone adrenocorticotrophic hormone, which is produced by the anterior pituitary. This differential stimulation reflects itself on the signaling cascade, so that the second messenger for ACTH is cyclic AMP, while the second messenger in case of angiotensin II is IP3, calcium, and diacylglycerol. These are the functional differences between the two zones, zona glomerulosa and zona fasciculata reticularis. We mentioned that cholesterol is the precursor of all steroid hormones. So where from the adrenal cortical cell gets cholesterol? There are two main sources for that. The first source is the lipoproteins, particularly LDL, and to some extent high-density lipoprotein. If you remember, LDL is full of cholesterol ester and is uptaken by all nucleated cells, particularly the liver. The steroidogenic cells, the adrenal cortical cells, uptake LDL. And this is one of the major sources of its cholesterol. If you remember, we mentioned that also high-density lipoprotein have a scavenger receptor on steroidogenic cells in the adrenal cortical cells and also in the gonads. So they are uptaken by these cells as well. But the major source is the cholesterol coming from LDL. Also, as we know, cholesterol can be synthesized de novo inside the cell. So we have these two main sources for cholesterol. Cholesterol in the cell is going to be esterified by ACAT, acyl-CoA cholesterol acyltransferase. This transfers acyl group from acyl-CoA to cholesterol. So cholesterol ester is synthesized while coenzyme A is released. This cholesterol ester is stored in vesicles or droplets within the cell. When the cell is stimulated, either by angiotensin II or by potassium ions, if it is in the glomerulosa, or by ACTH, if it is in the zona fasciculata reticularis, this cholesterol ester is going to be hydrolyzed by the enzyme esterase, and it will produce cholesterol again. Cholesterol, as we said, contains 27 carbons. And in order to produce any of the steroid hormones, be it in the zona glomerulosa or in the zona fasciculata reticularis. The first step and the rate-limiting step is the irreversible cleavage of 6 carbons from the 27-carbon cholesterol, and this is from its side chain. So we'll end up with 21-carbon pregnenolone. This step of side chain cleavage takes place in the mitochondria because the enzyme which carries out this cleavage step resides in the mitochondria. So cholesterol moves from the cytosol into the mitochondria in order for the cleavage step, the first step, to take place. Let us discuss this step of side chain cleavage in some details. This is cholesterol with its 4-ring structure. And then this is the side chain. At carbon atom number 3, we have the hydroxyl group. And there is also a double bond between C5 and C6. And of course, these are carbons 18 and 19. So this is carbon atom number 20 attached to carbon atom number 17. So if we count, this is carbon atom number 20. So this will be number 21. And then this will be number 22. So carbon atom number 17 is attached to carbon atom number 20. So this is cholesterol, and we are going to cleave this part. The enzyme system, which is responsible for that, is called side chain cleavage enzyme. And as we mentioned before, this is a mitochondrial enzyme. And this emphasizes the concept of compartmentalization in the biosynthesis of steroid hormones, because some enzymes are present in certain cellular compartments. This means that the substrate should move where the enzyme is in the compartment in order for the reaction or the step to be carried out. So the first step is the irreversible rate-limiting step of side chain cleavage, and the cleavage occurs between carbon atom 20 and 22. So the product is the 21-carbon pregnenolone and the 6-carbon isocaproaldehyde. Another name for it is 4-methylpentanal. So in the mitochondria, cholesterol with its 27 carbon in a reaction catalyzed by the side chain cleaving enzyme will cleave 6 carbons from the side chain between carbon atom number 20 and carbon atom number 22. And this produces pregnenolone, which is 21 carbons, and the 6-carbon isocaproaldehyde, or sometimes we call it 4-methylpentanal. This reaction takes place in the mitochondria and is irreversible and is an oxidation reaction. So we need oxygen, and also we need an ADPH as a source of electrons. And there are some intermediary enzymes and molecules. We have, for example, adrenodoxin reductase enzyme together with adrenodoxin. This adrenodoxin represents a shuttle for the transfer of electrons in the reaction. So in this step, cholesterol is cleaved between carbon 20 and carbon 22 by the side chain cleaving enzyme, which is a mitochondrial enzyme, in an oxidation reaction that needs oxygen and an ADPH, and produces pregnenolone and isocaproaldehyde. And we synthesize all steroid hormones from the 21-carbon pregnenolone. Now, let us dive a little bit deeper into the steps in biosynthesis of both mineral and glucocorticoids. At first, we are going to talk about some steps in a common pathway, both in mineral and glucocorticoids. First, start with some definitions. The pathway for mineralocorticoid starts from cholesterol and leads to the biosynthesis of aldosterone in the zona glomerulosa, while the pathway for glucocorticoid biosynthesis starts from cholesterol and ends with the production of cortisol and also corticosterone in the zona fasciculata reticularis. So let us first start with some key steps in the common pathway in the biosynthesis of both mineral and glucocorticoids. As we mentioned before, cholesterol is the precursor of all steroid hormones. So we start from cholesterol, the 27 carbon, with OH attached to carbon atom number 3, and the 8 carbon side chain attached to carbon atom number 17. The first step we have just described, the committed and the rate limiting step, which takes place in the mitochondria and which is catalyzed by the side chain cleaving enzyme, and the cleavage site is between carbon 20 and carbon 22. The result of the cleavage step, which takes place in the mitochondria, is the production of the 21 carbon pregnenolone, which, like cholesterol, still has an OH attached to carbon atom number 3 and still has a double bond between carbon atom number 5 and number 6 in ring B of the steroid nucleus. So the next step in the common biosynthesis of mineral and glucocorticoid is the conversion of pregnenolone to progesterone by 3-beta-hydroxysteroid dehydrogenase, which also has D5-D4 isomerase action. Pregnenolone is converted to progesterone by the action of the enzyme 3-beta-hydroxysteroid dehydrogenase on ring A and ring B. Remember, in pregnenolone, we had an OH attached to carbon atom number 3 like cholesterol. This is converted to an oxo group. The second action is an isomerase action. D5, D4 isomerase effect. The same enzyme does that job too. Remember, in pregnenolone we had a double bond between carbon atom number 5 and number 6 in ring B. Now, isomerization takes place and the double bond has moved to be between carbon atom number 4 and 5 in ring A instead. So the next step in the common pathway in the biosynthesis of mineral and glucocorticoid is the hydroxylation of progesterone at carbon atom number 21. This is catalyzed by the enzyme 21-hydroxylase, and this produces 11-deoxycorticosterone, which is known as DOC. DOC is an active sodium ion retaining mineralocorticoid, albeit weak. The last common step in the biosynthesis of both mineral and glucocorticoid is the hydroxylation at C11 This step is catalyzed by the enzyme 11-hydroxylase, which is a mitochondrial enzyme, like the sidechain cleaving enzyme. Another step that emphasizes the compartmentalization of the different steps in the biosynthesis of steroid hormones. So this last step signifies the hydroxylation at C11, thus producing corticosterone. So just to summarize the common steps in the biosynthesis of both mineral and glucocorticoid, we mentioned that in cholesterol, the side chain is cleaved between carbon 20 and 22 in a reaction catalyzed by the side chain cleaving enzyme, which is a mitochondrial enzyme, to produce the 21 carbon pregnenolone, which still has an OH group attached to carbon atom number 3 like cholesterol, and also still has the double bond between carbon 5 and the carbon 6 as in cholesterol. Then, in a reaction catalyzed by 3-beta-hydroxysteroid dehydrogenase, and this reaction takes place in the smooth endoplasmic reticulum in the cytosol, pregnenolone is converted to progesterone by the 3-beta-hydroxysteroid dehydrogenase, which converts the OH group, which is attached to carbon number 3, into an oxo group. Also, the enzyme possesses an isomerase function, thus converts the double bond between carbon 5 and carbon 6 in ring B to a double bond between carbon 4 and carbon 5 in ring A. So progesterone is produced that way. Then progesterone proceeds to be converted to 11-deoxycorticosterone, which is called DOC, by hydroxylation at carbon atom number 21. This reaction is catalyzed by 21-hydroxylase enzyme, which is also an endoplasmic reticulum enzyme. Then 11-deoxycorticosterone is going to be converted into corticosterone by hydroxylation at carbon 11, and this reaction takes place in the mitochondria. So now we have two mitochondrial enzymes in the pathway of the biosynthesis of steroids. We have the side chain cleaving enzyme, and we have 11-hydroxylase enzyme. So these two enzymes are mitochondrial enzymes, while the rest are smooth endoplasmic reticulum enzymes. Now we are going to talk about the biosynthesis of aldosterone, the mineralocorticoid, in zona glomerulosa. And in fact, we have now covered most of the pathway for the production of aldosterone in the zona glomerulosa by talking about the common steps in the biosynthesis of both mineral and glucocorticoids. So when we reach corticosterone, we are only 2 steps away from the production of aldosterone. And these 2 steps are catalyzed by aldosterone synthase, which is present only in zona glomerulosa and is not present in zona fasciculata reticularis. And this is why aldosterone is not produced in this combined layer of zona fasciculata reticularis. So, as I mentioned, we are only 2 steps away from aldosterone biosynthesis. So, corticosterone is acted upon by aldosterone synthase to produce first 18-hydroxycorticosterone. Aldosterone synthase, which has an 18-hydroxylase activity, it acts upon corticosterone and it forms 18-hydroxycorticosterone. The last step involves the oxidation of the ethene alcohol, ethene hydroxycorticosterone, its oxidation into an aldehyde, which is now called aldosterone. This reaction is catalyzed by the same enzyme, aldosterone synthase. We have to realize that aldosterone synthase is also a mitochondrial enzyme. So now we know of 3 enzymes which are mitochondrial enzymes in the pathway of the steroid biosynthesis. This includes the sidechain cleavage enzyme and the 11-hydroxylase, and now the aldosterone synthase. Of course, aldosterone synthase is synthesized in the mitochondria of the cells of zona glomerulosa. It does not exist in zona fasciculata reticularis. And this is why aldosterone is not produced in this zone, and the common reaction stops at corticosterone. As far as the biosynthesis of aldosterone is concerned, we have to notice that angiotensin II controls the cleavage step and the synthesis of ethene hydroxycorticosterone and aldosterone, the steps which are catalyzed by aldosterone synthase. So angiotensin II stimulates the conversion of cholesterol to pregnenolone, the cleavage step, and corticosterone to ethene hydroxycorticosterone and aldosterone. And this stimulation is via G protein-induced increase in IP3, calcium ion, and diacylglycerol.